Hello friends. In last few lectures we discussed about how the water and minerals are absorbed by the plant and how the water is transported to the leaves. Um, then this water and these minerals it is utilized by the plant in the process of the photosynthesis, and later on the growth of the plant takes place by utilizing the food material which is synthesized by the plant in the process of the photosynthesis. Now, these growth regulators, uh, uh, they play a very important role in the process of the growth of the plant. Today, we are going to discuss about what is growth actually, and how it takes place in the plant, and how the growth regulators they play an important role in the process of the growth. Now, when we are talking about the growth, that is particularly the plant growth, then we should study first of all the definition of the plant growth. What is this plant growth? The plant growth, it is the process in which it is an important physiological process in which the biomass of the plant it is increased. So increase in the biomass of the plant takes place. That process what we call it as a growth. When we define the plant growth, then the plant growth is a process in which the permanent and irreversible increase in the size, length, and volume of the plant takes place. Okay, permanent. It is defined as permanent and irreversible increase in the size, length, and the volume of the plant takes place. That process is called as growth. Okay, irreversible increase in the tissues, isn't size of the tissues, size of the organ, size of the what we call that leaves. When it takes place, that process is called as the growth. Now, where actually this growth is confirmed, there are three different main regions in the plant where the growth is confirmed, and these are the shoot apex, the root apex, and the vascular cambium. We know all that the root apex, it is present at the tip of the root. Shoot apex, it is present at the tip of the shoot, and the cambium. In all these three regions, there exists a particular tissue which is involved in the process of what we call it as the cell division. Getting it? It is continuously dividing. So that dividing tissue, what we call it as a meristem. At the apical site or at the shoot apex, the apical meristem is there. Root apex, there is also exist apical meristem, and to the cambium, there is a vascular cambium. Okay, and what we call it as that the cambium, there also exists the meristem that is called as the lateral meristem, by which the size or the radius of the stem of that particular plant it will be increased. So growth it is confirmed at these three regions: the shoot of the plant, then the root of the plant, and root apices of the plant, and the stem, the growth which takes place at by increasing the radius or the width of the stem. So at these three regions, there exist three different types of the meristem. Okay, so these because of this presence of this meristem, the growth is taking place in the plant because continuously in growing region, the cell division process is taking place continuously. Okay, now plant shows like other organism, plant also shows the growth curve, and we know that the growth curve, particularly the growth curve of the plants, that is which type of the plant, particularly the plant which are herbaceous in nature and the plant which are unicellular. In these plants, the growth curve is studied, and it is found that the growth curve in these plants is sigmoid. Sigmoid type of the growth curve is found. Here, we know that this sigmoid growth curve, there is initial phase where the growth is very, very slow. That phase is called as lag phase. Okay, this initial phase where the growth is very, very slow, and that initial phase where the growth is slow, that phase is called as lag phase. Then again, where the growth it is a increased, suddenly. Okay, and that phase of the growth curve is called as log phase, where the exponential, this phase is also called as exponential phase, where the growth rate is maximum. And again, it is a stationary phase where the growth is very very slow. This log phase, it consists of the vegetative growth of the plant. Okay, in this lag phase, the seedling stage. At this phase, the plant, it is in a seedling stage where the seed germination takes place and the growth is slow. The log phase, it is vegetative growth, vegetative period. And this, at maturity phase there exists the flowering stage of that particular plant, getting it. And at the stationary phase, uh, then at that time the growth of the plant is steady. Okay, and the seeds are formed, the fruits are formed from that particular plant, and the increase in the size and the volume of the plant, it is little bit stopped there at the stationary phase. Okay, and lastly the senescence and death will take place of that particular plant, getting it. So this is the growth curve. Okay, and this growth curve, it is same as that of the growth curve of a bacteria, and it is also shown by the growth curve. It is also shown by the herbaceous plants and the unicellular plants. Okay, and same type of the growth curve. This growth curve is nothing but what we call it as the curve which is drawn uh, by considering the time, and it is a actually it is a plotting of the graph. Okay, the graph which is obtained, or which is plot the growth rate of a plant against the time factor. When the graph is plotted by considering the growth rate of the plant and the time factor, then the graph which is obtained, that graph is called as growth curve, and which is S shaped, which is sigmoid growth curve in the plant, isn't it? Now there are certain what we call it the factors which affects the growth, and these factors, as we know that the food supply is very important factor. When the food supply is very less, the growth of the plant could not take place. Water, we all know that water is very very important, and when the water is minimum amount in a soil and it is not available to the plant, then growth is not possible. Okay, water supply. Then the temperature, we know that the temperature 33 is the 80, when the temperature is 28 to 33, then that temperature, it is a optimum for the growth, particularly in temperate the plants which are growing in temperate region. This temperature is different. The optimum temperature for the different plant is different. So the when the temperature, particularly the plants which are growing in the temperate region, then the temperature between 28 to 33 degrees centigrade is optimum for the growth. Okay, of the plant. Then oxygen supply, it is also important. Light, that light, it also the different factors, these are responsible for the growth, particularly the light quality, light frequency, and what we call it that the light intensity. All these factors, and one more factor that is called as the duration of the light, okay, which that plant receives. All these factors, the light frequency, light quality, intensity, what we call it, and the duration of the light, these factors are responsible on the growth. And the another important factor which is responsible for the growth is growth regulator. Now, let us see what is meant by the growth regulator. The growth regulator, these growth regulators are also called as the phytohormones. These are also called as the plant hormones, plant growth hormones. Okay, plant growth regulators, and these growth regulators, uh, which plays an important role in the growth and the development of the plant, particularly these growth regulators are uh, exist in the plant naturally. Okay, and they are present in the plant. They perform different functions in the plant. Okay, these growth regulators or these growth hormones, these are organic substances uh, which are produced in very minute quantities in the plant. These are also required in very minute quantities in the plant, and these are produced at one site of the plant, and then these are transported to different parts of the plant. Here, they are very effective for maintaining the growth of the plant, isn't it? Now, plant growth regulators, as we know that these are the phytochromes or phytohormones, what we call that. These are physiologically the function sites or functioning sites of these hormones. Uh, these are away from their synthesizing site. Okay, so they act. These are very active in very minute quantities, minute amount. Scientist Thiman in their of 1948 proposed the term phytohormones, as these hormones are synthesized in the plant, and the plant growth regulators include the auxins, gibberellins, cytokinins, ethylene, and abscesic acid. Okay, among these, some of the growth regulators or some of the hormones, they promote the growth, and the hormones which promotes the growth, these are called as growth promoters, and the hormones which inhibits the growth, some of them they also inhibit the growth, because the regulation of the growth in the plant, it is controlled by these organic substances, and the organic substances which are responsible for regulating the growth of the plant, those organic substances are called as growth regulators. These growth regulators are of different types: auxins, gibberellins, cytokinins, ethylene, and abscesic acid. Among them, some of them are growth promoters, which promotes the growth of the plant, and some of them are growth inhibitors, which inhibits the growth of the plant. And this promotion and the inhibition of the growth collectively or coordinately, we call it as a growth regulation. The regulation of the growth of the plant. Okay, so both the factors are essential. Promotion of the growth also essential, and the inhibition of the growth of the particular organ, it is also essential for normal growth of the plant. And this normal growth of the plant is achieved by both types of these hormones, that is called as the growth promoters and growth inhibitors. Getting it? Now, these growth promoters, first of all, the plant or the hormones which promotes the growth, these are called as the growth promoters. Okay. Now, plant growth regulators, these are defined. You can see here there are two definitions of the plant growth regulators. Okay. Let us see one by one. It is defined as the organic compounds other than the nutrients that affect the physiological process of the growth and development in the plant when applied in very low concentration. These are called as the growth regulator. Another definition of the growth regulator, it is the growth regulator. These are defined as either naturally or synthetic compounds that are applied directly to a target plant to alter its life process or its structure to improve the quality, increase yields, or to facilitate the harvesting. So, for any one of this reason, when these organic compounds are applied, then these organic compounds are what we call it as a growth regulators. Okay, now the plant hormones, incorrectly used, it is restricted to naturally occurring plant substances. They fall into the five classes as we discussed. These are auxins, gibberellins, cytokinins, abscesic acid, and ethylene, and of which the some are growth promoters and some are growth inhibitors. The plant growth hormones, primary site of the action of the plant growth hormones at the molecular level remains unresolved. Okay, because these are the reasons. Okay, and let us see. These are the five types of the what we call it, or five groups of the growth hormones. Okay, the first is auxin. The auxins, these substances generally resemble the indoacetic acid, and they have ability to stimulate the elongation of the coleptides. Auxin, we consider it as a growth promoter. Gibberellin, we also consider gibberellin as a growth promoter. And the cytokinin, we also consider the cytokinin is also a growth promoter. Okay, so these three, auxins, gibberellins, and cytokinin, these three are the growth promoters. Okay, and the role of the cytokinin we all know. Usually, these substances we will see in detail later on, but these substances are made up of the adenines. Okay, then let us see inhibitors. The inhibitors are the regulators which inhibits the growth, and particularly these inhibitors are ethylene and abscesic acid. So ethylene and abscesic acid, these are considered as a growth inhibitors. Now let us see one by one the auxins. The first, that is called as the auxin, growth regulator, first growth regulator, auxins. So these auxins are a group of phytohormones which are produced in a shoot and in a roots, particularly the apices of the shoot and root, and they migrate from the apex to the zone of elongation. Okay, the auxin promote the growth, so these are considered as a growth promoters, and the promotion of the growth is along the longitudinal, what we call it as a the longitudinal axis. Okay, the longitudinal axis of the plant, and hence the name is called as auxin. The term auxin was introduced by a scientist Kogel and a scientist Smith in the year of 1931. It was scientists went who isolated the auxin from the seeds of the avena in the year of 1928. Okay, he isolated the auxin from the seeds of the avena coleoptile. Particularly, he isolated these auxins from the tips of these avena coleoptile, and these avena coleoptile. Uh, it is found that on curvature test, these auxins are isolated, and these auxins are responsible for elongation of this particular plant. Okay, so the plant auxin are widely distributed in the plants throughout the plant. However, the abundant in the growing tips, such as the coleoptile tips. It means that the auxins were present in all over the plant, throughout the plant, but their concentration, it was more at the tip or at the growing tip of that particular plant. Okay. The Indoacetic acid, that is IAA, it is the only naturally occurring auxin in the plant. Okay, and it is available in abundant quantity, abundant amount in the plant. This is the avena curvature test. Okay, here you can see the cut of the tip of the avena coleoptile. When these are placed on the agar plate or agar block, then auxin, it is diffused into the agar block. Okay, we can see here this agar plate here, it is a yellowish or cream colored agar plate is there. Later on, when we place these the tips of the avena coleoptile, then we found that it becomes blue colored. Okay, and this is because of the distribution of the auxin into the agar plate. Okay, it proved that the auxins are present at the tip of the what we call that the coleoptile. Now, these auxins are exist, as we discussed here. The auxins are exist in different forms. The main and important form of the auxin is IA, that is indole acetic acid. Then it is also exist in the form of IBA, indole butyric acid. It is also exist in the form of NAA, naphthalene acetic acid. Then MCPA, 2-methyl 4-chlorophenoxyacetic acid. Then TIBA, 2,3,5 tri-iodobenzoic acid. 2,4-D, that is 2,4-dichlorophenoxyacetic acid. 2,4,5-T, that is 2,4,5-trichlorophenoxyacetic acid. Okay, so naturally auxins may occur in the form of free auxins, isn't it? Either free auxins which freely move or diffuse out on or diffuse out of the plant tissues radially, or in the second form of the auxin, which is found to be plant, it is in the bonded form, what we call it as the bond auxins, which are released from the plant tissues only after hydrolysis, or only after autolysis, or only after enzymolysis. So there are two forms: one auxin, which is available, which is present in a free form, and the another which is present in the bonded form. Okay, now let us see the physiological actions of the auxins. We know that the main and important function of the auxin, or the function of auxin, it is a what we call it as the elongation. The before that, let us see how these auxins they help, or the action, or the mode, or mode of the action of the auxin. Okay, now these auxins, we know that these are synthesized at the tip of the root and shoot, and later on they are transported to different parts of the plant. When they are transported to different parts of the plant, then at that time the auxins are binded. How we are studying how the auxins are what we call that uh, they perform their role, or how they perform their action in the plant. There are three different steps and main steps. These are main steps which are involved in the activity of the auxin. And these three main steps are: first, auxin binding, then the induction of gene expression, and the third stage is its function, that is called as cell elongation. Okay, so the auxin binding, that is IAA, binds with the auxin binding protein. Okay, indoacetic acid, what we call the auxin. First of all, the auxin binds with the auxin binding protein, that is called as ABP1, auxin binding protein, on the surface of the target cell. These ABP first, these are present on the surface of the target cell, and these auxins are binded to the ABP first, that is auxin binding protein first, and later on this protein it transport the auxin into the cell, isn't it? When the auxin entered in the cell, then the another important role, or the step in the auxin action, mechanism of the auxin action, it is a uh, auxin's action, it is the induction of the gene expression. The IAA, indoacetic acid, induced growth appears into the tissues after minimum lag period. Okay, that is 10 to 20, or particularly 12 minutes. Okay, so IAA induced growth appears in tissues after a minimum lag period. So this IAA induced growth, uh, in this phase particularly, the lag period is reduced. Getting it? The lag period is reduced in the plant. We have studied the sigmoid curve. So because of the induction of the indole acetic acid, the lag period, which is very slow, that slow period of the plant is reduced, and afterwards suddenly the log phase, it is coming in the sigmoid curve. So when the period of the lag phase it is reduced, suddenly the log phase will come, and the log phase will come very soon, very early in the plant, and suddenly the elongation of the cell, it will take place. And lastly, the last step is the cell elongation, which occur in the mode of the action of the enzyme. So physiological effect. While studying the physiological effect of the auxin, the first we discussed here already, that cell division and the cell elongation. The primary physiological effect of the auxin, no doubt, it is the cell division and the cell elongation in the shoot particularly, and in the root as well. It is important in the secondary growth of the stem. Okay, stem and the differentiation of the xylem and the phloem tissue. So the cell elongation occurs in the log phase. Okay, which is um, uh, the phase of a long duration. Okay, but here the within a short time, this in this phase, the elongation of the cell takes place for 18 to 20 hours. The log period is there. The IAA induced accumulation of the IA into the cell, it reduces the lag phase, and the log phase comes very early in the cells. Okay, and because of that, the cell elongates very fastly. Okay, so the another effect of the what we call it as the auxin, it is the apical dominance. We know that the apical dominance is the that type of the phenomenon in which the growth is dominated to the apical site only. In many plants, if the terminal bud is intact and it is growing, the growth of lateral bud is just below remains suppressed. Okay, many plants show such type of the phenomenon or such type of the what you call that behavior. Here, the growth of the plant it is at the apical site only. Okay, and the lateral buds do not grow. Okay, the apical bud grows only. The removal of apical bud, it results into the rapid growth of the lateral bud, isn't it? The removal, when we remove the apical bud, it results into the lateral uh, rapid growth of the rapid growth of the lateral buds. This phenomenon in which the apical bud dominates over the lateral bud and does not allow to grow the lateral buds, isn't it? This phenomenon is known as apical dominance phenomenon. And this apical dominance phenomenon in plant, it is because of the presence of these genes or growth regulators, that is called as the indole acid. The scientists Cook and Thiman in the year of 1948, he pointed out that this scientist pointed out that the apical dominance might be under the control of the auxins, which are produced at the terminal bud and which is transported to downward through the stem. The lateral buds and the hinders the growth. The lateral buds of the that particular plant, these lateral buds undergo the suppression of the these hormones, hormonal effect, and the because of this more amount of the hormones are present at the apical side, the apical bud grows faster. Okay, they removed the apical bud and they replaced it with agar block. This resulted in the rapid growth of the lateral bud. But when they replaced the apical bud with agar block containing the auxin, the lateral buds remain again in suppressed condition and these lateral bud did not grow. Hence, it proved that the apical bud dominance, it is because of this phenomenon, apical bud dominance phenomenon, in which the apical bud grows faster and which suppress the growth of the lateral bud. This phenomenon is because of the presence of what we call as the auxins to the apical site. Okay, you can see here. This is the apical. In first diagram, we can see the apical bud. It is growing, but when we cut the apical bud, then the lateral buds are also starts to grow. Okay, so the growth of the lateral bud it begins when we cut the apical bud or when we remove the apical bud. Okay, and this is apical dominance phenomenon. Now, next is the root initiation. In a contrast of the stem here, it is a different from the apical bud dominance. In stem, what we have found that when the auxins are present in the stem, these are present in apical bud. The growth of the apical bud is promoted, but in case of the roots, the growth of the root, it is not promoted due to the presence of the auxin. Okay, auxin inhibits the elongation of the roots, but the number of lateral roots are formed in more amount due to the presence of the auxin in the roots. Okay, so lateral roots are formed in more amount, whereas the elongation of the root, it is not promoted due to the presence of the auxins or indoacetic acid. The higher concentration of the auxin induces more lateral branches of the roots. The apical or the particularly the application of the indoacetic acid in a lanolin paste. Okay, that when we cut the young stem results in an early extensive rooting This fact is of great practical importance and has been widely utilized to promote the root formation in economically useful plants, which are propagated by cutting method. Okay, so next importance is the what we call the prevention of the abscission. We know that the abscission is the process here, the falling down of the leaves and falling down of the flowers, falling down of the fruits takes place. And this abscission process, it is the process at which the abscission zone is formed in the petiole or in to the stalk of the flower or stalk of the what we call that the fruit. And once the abscission zone is formed, uh, then that detach that particular organ, it will be detached from the axis or that plant axis, hitting it. It may be branch or whatever it may be. So this process of the abscission or formation of the abscission zone, it is uh, inhibited in presence of the hormone auxin. Okay, natural auxin prevents the formation of the abscission layer, which may otherwise result in the falling down of the leaves or flowers or the fruits. Now, the parthenocarpy, we know the process of the parthenocarpy, where the formation of the fruits takes place without forming the seeds. The formation of the fruits without forming the seeds is the process what we call it as the parthenocarpy. Auxin can induce the formation of the parthenocarpic fruits. Okay, that is a for formation of the fruit without pollination. As pollination is not there, seeds will not be formed. Okay, and fertilization is absent, the seeds are not formed. So the formation of such type of the unfertilized seeds or the seeds, the fruits without seeds. This process it is promoted by the auxin. Okay, in parthenocarpic fruits, the concentration of auxin in the ovaries is higher than the in the ovaries of the plants which produce the fruits only after the fertilization. Okay, so in the later case, the concentration of the auxin in ovaries increases after the pollination and the fertilization. It is found that after pollination and the fertilization, the concentration of the auxin it is increased. But after fertilization, if the concentration of the auxin it is increased, it will not allow to form the seeds in the what we call it that particular fruit. Now the respiration, the auxin stimulate the process of the respiration, and there is a correlation between the auxin induced growth and the respiration. Auxin may increase the rate of respiration indirectly through increased supply of ADP by rapidly utilizing ATP in expanding the cells. Okay. Now, callus formation, the callus, it is undifferentiated mass of loosely arranged parenchymatous cells, and this callus formation, it takes place when the wounds are formed to the stem of the plant. When the wounds are formed, their um, growth of the tissue takes place, and that growth of the tissue, it is induced because of the increased concentration of the auxins. These auxins induces the growth of the tissue, and that tissue newly formed mass of the loosely arranged parenchymatous is it is nothing but the callus. It is later on it develops, but previously or in early stage it is the callus. So the callus formation, that is a loosely arranged parenchymatous cells, the mass of loosely arranged parenchymatous cell, the formation of this callus, it is also promoted by the auxins. Now, the eradication of the weeds. Some synthetic auxins, especially 2,4-D and 2,4,5-T, are useful in eradication of the weeds at higher concentration. When we use these 2,4-D and 2,4-T at higher concentration, we apply it on the plants, but we apply or we spray it on the weeds. Then what happens? The uncontrolled cell division takes place in the plant. These auxins are absorbed by the leaves of the plant, and these are transported to the apical meristem. And here the uncontrolled cell division takes place, and as a result of that, the plant will die. These leaves also dries because of the plasmolysis. So the drying of the leaves will take place, and dying of the plant it also takes place because of the uncontrolled cell division. It results into the death of that particular plant, and it results into the eradication of the weeds. So auxin also helps in the eradication of the what we call that the weeds. Okay, the flowering and the sex expression, auxin generally inhibits the flowering, isn't it? But in pineapple plant and in lettuce plant, it promotes the uniform flowering. Okay, in some plants, the auxin promotes the uniform flowering. Distribution of the auxin in the plant, we know that, and we already discussed it that in plants, auxin, that is indole acid, is synthesized in growing tip of the meristem or the meristematic region. From here, it is transported to the other parts of the plant, and the higher concentration of the indole acid is found in growing shoot tips, the young leaves, and the developing auxiliary shoots, isn't it? In monocotyledon seedling, the highest concentration of the auxin it is found in the coleoptile tips, which decreases progressively toward the bases. Okay, and this shoots, the here the growth is continuously taking place. Because of that reason, the concentration of the auxin is higher in the um, tip of the plant. Okay, in dicot seedling, the highest concentration it is found in a growing region of the shoot, obviously. Okay, and this indole acetic acid, the biosynthesis of the indole acetic acid, it is discussed, or it is experimentally proved that the it is found that the amino acid tryptophan, it is actually responsible, or it is a precursor. Tryptophan, it acts as a precursor for the synthesis of indole 3 acetic acid. So the tryptophan, it is actually primary precursor for the synthesis of the indole acetic acid. Okay, and by elimination process, the tryptophan to form the indole 3 pyruvic acid, followed by the decarboxylation to form the indole 3 acetaldehyde. The enzymes involved are tryptophan deamination and idole pyruvate decarboxylase respectively. So these two enzymes, tryptophan deamination and indole pyruvate decarboxylase, these two enzymes are responsible for uh, conversion of the tryptophan into the indole 3 acetaldehyde. Okay. Now later on, by the process of the decarboxylation, the tryptophan to form the what you call that tryptamine, followed by deamination to form the indole 3 acetaldehyde. And the enzymes involved are the tryptophan decarboxylase and tryptophan oxidase respectively. So the indole 3 acetaldehyde can radially be oxidized to indole 3 acetic acid in the presence of the indole 3 acetaldehyde dehydrogenase enzyme. Here, the indole 3 acetaldehyde, which is formed in the previous step, it is converted into the indole 3 acetic acid. Okay, so this is how the synthesis of the indole acetic acid it is taking place in the plant, and this is the overall reaction of what we call that indole acetic acid. Indole acetic acid plus H2O2 plus O2, it results into the formation of 3-methyl oxidase or oxidol plus H2O plus CO2. This is how the indole acetic acid are destructed, isn't it? Destruction of the indole acetic acid or inactivation of the indole acetic acid takes place in the plant, and this destruction or the inactivation is also important for controlling the growth of the plant. Since these indole acetic acids are destructed, further cell elongation or further cell division or further growth of the plant could not take place. Okay, and this mechanism of the mode of the action we have studied already, how the indole acetic acid it works. Okay, so this is all about the indole acetic acid. In next lecture we will discuss about the gibberellins, the another type of the growth regulator, and this indole acetic acid, it helps, mainly it works for what we call it as the elongation of the cell. In next lecture, we will discuss about the gibberellin. Thank you very much. Thank you. Thank you, one and all.